Buenos días a todos, señor rector de la Universidad Autónoma, presidenta del CSIC, compañeros del Centro de Biología Molecular y demás científicos y amigos que estáis viendo este acto por streaming. Querida Lucía, pues os doy la bienvenida a la primera lección conmemorativa, Margarita Salas. Es increíble, pero ya ha pasado un año de su fallecimiento y en su honor pues instauramos estas lecciones conmemorativas. Agradecemos al CSIC la instalación de un busto en homenaje a Margarita Salas que acaban de inaugurar el rector y la presidenta en el hall del CBM y que servirá pues de referente y de recuerdo de Margarita Salas, nuestra compañera en el CBM, donde desarrolló su trabajo durante 42 años. Y bueno, va a estar siempre ahí para servir de referente también a todos aquellos científicos, incluso cuando ya no estemos en activo los que la conocimos trabajando aquí, pues siempre estará ahí ese gusto para servirnos como de ejemplo. La verdad es que se hace un poco raro esta lección conmemorativa con la sala medio llena. Por supuesto, en otras circunstancias hubiese estado lleno, no solo por Margarita Salas, sino también por la calidad de nuestra conferenciante hoy, a quien agradezco su presencia aquí, Marisol Soengas. Y sin más, cedo la palabra al rector de la Universidad Autónoma, Rafael Garese.
Muchísimas gracias, Lourdes. Buenos días a todos, querida presidenta, querida directora, querida Lucía, Jesús, todos los amigos y amigas del CBM. Para mí, desde luego, como rector de la Universidad Autónoma de Madrid, pues es un privilegio poder compartir hoy este momento con todos vosotros. La verdad que la figura de Margarita Salas es una figura que ha tenido un impacto en la ciencia española extraordinario y por muchísimos motivos, ¿no? El primer motivo, evidentemente, es por la calidad del trabajo que ha desarrollado, de primerísima línea internacional. El segundo es porque ha sido capaz, y no hay muchísimos ejemplos en nuestro país, de crear una escuela de científicos también de muy alto nivel y que han ya seguido su camino y ocupado las posiciones que tienen que ocupar en distintos centros de nuestro país, ¿no? Y aquí mismo en el propio CBM, y eso es también muy de agradecer, porque muchas veces los grandes científicos a veces no dejan detrás una escuela y Margarita ha sido capaz de crearla. Desde luego, para la Universidad Autónoma de Madrid, Margarita pues, ha sido siempre una referencia que estaba en el CBM, que fue una apuesta que se realizó muy innovadora en su momento, ahora que se habla tanto de innovación, muy valiente en su momento, en la cual una universidad que casi empezaba en aquellos años y una institución como el CSIC, pues llegaron a hacer una propuesta de centro mixto que luego ha sido ejemplo para muchísimos a lo largo del tiempo y en muchísimas universidades, y para nosotros como universidad autónoma pues marcó un rumbo y una diferencia extraordinaria en lo que es la evolución de la misma. Bueno, pues en este momento de la creación del Centro de Biología Molecular, pues hubo una serie de científicos, de científicas que jugaron un papel determinante en el mismo, y entre ellos, desde luego, estaba Margarita, estaba Eladio también, y se integraron en la docencia que se empezaba en aquellos momentos en el área de biología, de microbiología. Desde luego, fue una época que yo siempre la recuerdo. Yo no la viví en primera persona, pero no tardé mucho en incorporarme a la Universidad Autónoma y todavía se respiraba ese ambiente creativo, vibrante, fresco, que daba un aire completamente diferente. Y esto fue gracias a esta apuesta y gracias a la categoría extraordinaria de las personas que se fueron incorporando en aquellos momentos, y entre ellas, desde luego, Margarita ocupó una posición muy relevante. Posición muy relevante que ha perdurado en el tiempo, afortunadamente, realizando un trabajo enorme y siendo un ejemplo de buena ciencia, pero también un ejemplo de científica, de mujer dedicada a la ciencia. En este problema tan importante que tenemos de género en nuestro país, en Europa y en todo el mundo, pues Margarita ha sido una referencia que ha abierto camino y que creo que la situación poco a poco va cambiando, pero precisamente gracias a personas como ella. Es decir, que para mí como rector simplemente agradecer al centro. Desde luego, la obra del artista Lusto es precioso. Me parece que es muy elegante, muy bonito y que también va a ser, digamos, pues bueno, un recuerdo permanente para el centro y para los jovencitos que no hayan conocido a Margarita, porque los que hemos tenido la fortuna de conocerlo nos acordaremos de ellos toda la vida, ¿no? Porque gracias a personas como ella, pues las instituciones progresan y avanzan y su recuerdo ya es imborrable. Eso es lo bueno que tiene dedicarse con intensidad a un trabajo y a una institución. Que en este caso han sido dos, el CSIC, desde luego, que es su alma mater, pero también nuestra universidad, que siempre la ha considerado un referente y una más de nuestra familia de este campus. Así que muchísimas gracias por la invitación y, como decía al principio, para mí ha sido un privilegio compartir estos minutos con todos vosotros. Muchas gracias.
Directora del Centro de Biología Molecular, Severo, Ochoa, Lourdes, rector de la Universidad Autónoma de Madrid, querido Rafael, vicedirector del Centro de Biología Molecular, Jaime, Jesús, Lucía, compañeros y amigos, muy buenos días a todos. Es un día entrañable, porque homenajeamos, en mi caso personal, a una compañera, a una científica y además a una buena amiga. Y como decía Rafael, parece mentira, ¿no?, que ya haya transcurrido un año. La verdad es que parece que sigue ahí. Y ese busto que tenemos ahí en la entrada va a hacer que permanezca y que esté presente en el recuerdo. No la vamos a olvidar, desde luego, pero hacemos que Margarita continúe con nosotros. Y también Soledad Soengas, Marisol Soengas, que inauguras esta primera lección conmemorativa y que estoy convencida y espero que desde luego que tenga continuidad, y es uno de los muchos actos que tenemos previsto organizar para perpetuar la presencia de Margarita, la figura de Margarita. Tenemos muchas iniciativas en marcha. El mural también del Centro de Investigaciones Biológicas, Margarita Salas, CIB. Nuestro tan deseado homenaje, que ya hemos pospuesto 2 veces, pero esto no quiere decir que no lo vayamos a hacer, porque queremos hacerlo y con toda la fuerza y con toda la presencia que requiere, pues, la figura de Margarita Salas. Estamos trabajando en la creación de la Fundación Margarita Salas, que yo creo que también es un esfuerzo importante y tiene un valor de continuidad en el tiempo de la científica, de la persona. Y en todo ello estamos, desde luego, como muy bien decía el rector Rafael, codo con codo, la Universidad Autónoma de Madrid y el CSIC. Pienso que Margarita nunca va a desaparecer en nuestras memorias, pero que cualquier contribución que podamos hacer, aunque sea pequeñita, van a ir sumando y vamos a hacer que a esta mujer que ha roto fronteras, que ha roto barreras en una época difícil, como la que vivió, que es un referente en todos los sentidos, como decía, va a estar presente. Aquí la vais a ver todos los días cuando venís a trabajar, cuando vengan las visitas y luego en el mural que también tenemos previsto para el CIB. Solo me queda, pues, de verdad, agradeceros a todos y me encantó lo que has dicho, Lourdes, en una sala medio llena. Vamos a ver la sala medio llena y no medio vacía, porque yo tengo que confesar que sigue costando hablar, ¿verdad? Para pocas personas y además con las mascarillas. Pero bueno, ya está, tenemos que acostumbrarnos, aunque nos cueste y resulte difícil. Y quiero, desde luego, desearos una buena jornada. Marisol, muchas gracias por estar aquí con nosotros, que Marisol también es un referente auténtico, y por colaborar en esta jornada. Muchas gracias a todos y mucho ánimo, ¿verdad? Que tenemos que tenerlo.
A continuación, pasamos a la parte científica del acto, lo que es la lección conmemorativa Margarita Salas, y el profesor Crisanto Gutiérrez hará la presentación. Y ya pasamos al idioma inglés.
Okay, so good morning, everyone. I would like to start just saying that only a couple of days ago, we would have been celebrating Margarita's birthday. And I think that one of the best presents that we could give her is to celebrate her birthday with the first memorial lecture in her name. We are all really appreciating the organization of this memorial lecture by the CBM, and I'm pretty sure that the situation will continue for the years to come. Today, we are also, I'm particularly very pleased to introduce our today's speaker for this memorial lecture, Marisol Soengas. Marisol came from A Coruña to finish her degree here at the university, and then joined Margarita's lab for the PhD, obtained in the middle 90s. And I also, I was very lucky in those days because we shared a lot of work in the lab at this time. After that, in 1997, Marisol decided to switch gears and start working in a completely different field. And she moved to the Cold Spring Harbor Laboratory and joined the lab of Scott Lowe, where she started to becoming more and more interested in biomedicine in general. And in particular, she made very, very strong discoveries in the field of apoptosis. And after, based on her performance, during those years, she obtained a position at the University of Michigan in Ann Arbor, and joined the Department of Dermatology in 2002, if I remember correctly, Marisol. And at that time, she started a very, very productive lab in the US. She obtained a number of different awards, the Lifetime Award from the University of Michigan, and a few others from different foundations. And in 2008, I think that the Spanish science community was very lucky because the CENIO was able to recruit her, and she launched a very important, I think it's a reference worldwide, laboratory of melanoma research. And she's now developing new technologies and new concepts, I think, in the field of melanoma research, especially in the initiation, progression, metastasis, and so forth, which I think will be the subject of her talk today. Marisol has also been awarded with a number of awards, from different foundations and from the US and also from Europe, including a recently awarded ERC Advanced Grant, for example, and many others, and a few prizes, of course. In particular, I think that she is particularly happy having received the Josefa Bonenburger from the Junta de Galicia. And without any further delay, I would like to pass the word to Marisol, and I'm sure that we will enjoy her talk. So thank you for coming, Marisol.
Okay.
So, well, thank you so much, Crisanto, for the presentation. And it's always a pleasure to be here at the CBM It's also a pleasure to see people, even if it's half full. And in particular, it's very emotional for me today because we are honoring Margarita Salas, who has trained so many people and has impacted so many people here at the CBM, in Spain, and abroad. So really, I'm going to start the presentation. Let me see. This is now? Now should be no? Or is not? Yes, yes. Okay, so I mean, we just heard and so how important, and of course the concept of a family that Margarita created here at the CBM, but also how important she was elsewhere. And I thought to start with this slide that I think she would have liked. She would have liked the message, and I'm sure she would have liked what is portrayed there. And this is dream with ambition, lead with conviction. And this is what Kamala Harris said when she became vice president of the US. And I think this lead with conviction is definitely what Margarita has, I mean, so it's her motto, I would say, and also the concept of moving forward. But I like this picture also because, so this is a composite. And here on the left, so it was portraying Ruby Bridges, who she was the first female student going to, it was a segregated school, and she had to be escorted by the police. But this action so impacted so many people afterwards. And then in the left, you see here Kamala Harris, so for example, people like her getting into leadership positions, but also the fact that Kamala Harris is going to also influence a lot of women for the future. And if we think about Margarita, I think it's the perfect image because she has influenced men and women, myself as well, and many others. So I think this is a good image for her. And then the sculpture that we have outside, I think is actually a great image to keep remembering her. For me, I mean, I'm not going to go too much on this because this was supposed to be a scientific presentation, but of course I had to show some pictures of what it was personally for me being part of her group. So in the left, so so Crisanto mentioned, so I joined the lab when, Margarita's lab when I was in the finishing biology. And this was one of my, a meeting in Keystone, one of my first international ones. And it was very nice to see also and to learn so how she was, not just in the lab, but outside. We have kept in contact for many, many years. This is here in the middle when she got the Medalla Che Garay. So, and then here in the left more recently, the CNIO had an event for science, and then so was also portraying her image. So it was very nice. And so just so a few things of when I being asked about Margarita, what I always say. So I think on the one hand, I envy really her ability to pick the right people. So to identify talent and to get people to join her group. And then I think if we ask, so we asked anybody that worked with her in the lab, so what was to be in Margarita's group, I think we will always say rigor. So it's like the scientific rigor, right? So this, I think we were like trained and really was engraved in us that we have to repeat, that we have to quantify. And importantly, I think, and she had mentioned this in several interviews that she had, honesty and scientific honesty. And this is very important nowadays that you see a lot of people selling stories. And with Margarita, the important point were the data, and the data had to speak for themselves. And so, well, I could go on and on, so many other things that I thought important, or I think that was important from her that I learned. But I think another point that I think part of why the lab was also very successful is because she knew how to delegate. And delegating is very important, particularly if you are very high up. And the way the lab was organized with senior people that would take care of students. So this point of nobody left behind. I think when you knew that you got into Margarita's lab, you would know that you would finish, and you would finish with publications and in good way. So at my time, when it was like in the 90s, mid-90s, is where the senior people, the investigators that we have, Luis Blanco, Crisanto Gutierrez, Fernando Rojo, and Jose Miguel Hermoso. And I think we have to also be very thankful to all of them for their support. Myself, and I'm going to get just a few slides on what it was my tenure through the lab. So I got really very lucky. I just, you know, it was in the fourth year in biology, got in. And so this was a project that Luis Blanco was leading. I learned a lot from him. Maria Blasco, that you see, is here now, the director of the CDM. You see this guy here, Esteban. And then, so it was my first paper. Second paper was then, first he helped me, then I helped him more in the exo activity, and the rest is history. We have here part of the lab at that time, and so very productive, I would say, interaction on many different levels, of course, personal-wise. And then for my thesis, I was very fortunate, I have to say. And I was fortunate for many reasons. So to having both Margarita and Crisanto supervising this work, it's not that we worked together, it's like Crisanto really was so important to me, because he was one of the first things that he told me, I think the first day when we met, and said, you know, today I'm telling you what you want to do, but tomorrow you have to propose the experiments to me. And this is something that I always keep to my students, this thing that I had to propose experiments, I have to propose experiments. And so we got into a very biophysical studies. I'm impressed when I see the figures, actually, because there were a lot of techniques that I had to develop, and we had a lot of fun there. And this is my thesis, and now that there's a lot of computational biology, so it was this Crisanto and Jose Antonio worked in this program, FluoCUFIT. Anyway, so even, and this I also tell the students, sometimes you might feel that there are projects that are very complicated, but you know, really there are very few things that are impossible. And in science, we try to go for the possible. So this is kind of my summary of what was for me to go through Margarita's group. And so Crisanto mentioned I'm from La Coruña, Luis Blanco is here. So, and I think for those of you that have been in Galicia and in general, so I'm sure that you know one of our iconic monuments, the Torre de Hércules, is of course a World Heritage Monument, and is the longest standing lighthouse that is still active. It's from the Roman time. But when I liked this picture, well, first because it's from La Coruña, I think I like it, but also because it is also, in a way, reflects some work that we're trying to do in my group in terms of visualizing the process of tumor initiation, progression, and metastasis in melanoma, but you will see that it's applicable to other tumor types as well. So I'm going to very, very briefly touch upon also what melanoma is. Then I will tell you about a series of mouse models that we have developed, and we call them MetaAlert. And the MetaAlert goes about this concept of imaging and in vivo. So how using these mice and also patient biopsies and large data sets, we trying to discover new mechanisms of progression. I'm going to tell you then, as well as how we are getting into mechanisms of immune modulation and indeed immune suppression. And I will end up with treatment, and also telling you that, yes, from basic research, you can get into translational research. It's a company that we co-founded and is now in phase 2 clinical trials. To get started, so the theme of my group is melanoma and lesions that are derived from alterations of melanocytes. So now we have melanocytes in very different organs, but most of the melanocytes are in the skin. Here you have a representation. I hope you see the pointer. So melanocytes, in normal skin, you have them here, these cells, and they are individually placed in between keratinocytes in the basal layer of the epidermis, okay? And these are the cells, are very important. These are the cells that are responsible for pigmentation. They get activated. They are named melanocytes because they produce melanin, and this is important for pigmentation. But so these cells, because they are in the skin, they receive and respond to UV radiation. They get altered very frequently. And all of us, we have alterations of melanocytes. And these are the nevi, the lunares, okay? So we see these nevus because as you see here, there are two, all of us, we have them. So what they are, we see them because they are groups, lots and lots of melanocytes. And they keep more or less in the interface between the epidermis, the upper layer of the skin, and the dermis. And so this is benign, and they are benign because they, at some point, they proliferate, but they stop. So when we talk about mechanisms of tumor suppression, really, so nevi are one of the best examples of things that started to be wrong because all these cells have mutations, the nevi cells, but then they are stopped. And on the other hand, melanoma is the bad side of this. So it's a situation where cells, these melanocytes, keep proliferating, they invade the epidermis. That's not so problematic, but the problem is that very early on and from very thin lesions, they can invade and can give rise to metastasis. So many groups, so still there is a lot to understand how the nevus are initiated, how they stop, and then why the melanomas progress. It's very complicated. So you look at yourself or your skin, you will see that you have different types of and sizes of these nevi. And indeed, there are many types of them, the same way that there are many melanomas in you see here, these ugly pictures, but you can see in the skin, mucosas, in the eye, ocular melanomas, pigmented, non-pigmented. So it's a lot still in terms of to be identified in terms of progression and also diagnosis. In my group in particular, so we have various areas of research. On the one hand, so we are interested in the fingerprint. So what defines the lesion, that it can be just barely millimeters in depth, can go and metastasize. So really what makes melanoma so aggressive and so metastatic. The other is going, so okay, it progresses and metastasizes. Typically, melanoma patients present with metastasis in various and many different organs. So why is that? So what are the drivers, and can we identify vulnerabilities that we could target therapeutically? So I'm not going to get into details. I'm going to talk a little bit later on mechanisms of, so in trying to define this fingerprint of vesicular trafficking and how we are using mechanisms of the cells has to respond to stress, to modulate metastasis. I'm not going to talk about this, but we also have kind of several studies in the lab trying to identify these bad roots of the metastasis process. What I'm going to talk more is about this, the models I've mentioned that allows to visualize this early, like the metastasis process. I will talk about this compound, Bio-110, that was the Bioncotech Therapeutics, and how it's been developed further. And we're very happy. So in a few days, we're going to have this cover or something like this in Nature Medicine, and it's how we're trying to also identify proteins that mediate metastasis, and we think it could be good targets for therapeutic development. Okay. So now getting into the concept of imaging. So why, and I mentioned why we are so interested in visualizing. We're interested in visualizing this process because I told you that, so this is a melanoma. So melanocytes are here on the left. This is a melanoma, and it may look big, but this is millimeters, okay? It could be 1 millimeter, 1.5, and it has already the ability, these cells, to intravasate through the vasculature. Typically the lymphatic vasculature, also the angiogenic vasculature. And melanomas are very efficient into this. They're also efficient at bypassing recognition and attack, what is the immune surveillance in the lymph nodes. And then when they are very aggressive, as I mentioned, they go and disseminate everywhere. So we know lots of melanoma has been one of the, well, many tumors, but melanomas in particular. So the identification of large amounts of mutations and epigenetic alterations. Indeed, melanoma is the tumor that has the largest number of mutations that have been described. And we know a lot, but I think still this concept of how the crosstalk between the tumor cells and the vasculature is organized is not very clear, and then we still have things to do in terms of treatment. So in the concept of imaging, so what we wanted to image, so was this interplay or between the tumor and the vasculature. And we interested in the lymphatic vasculature because lymphatic endothelial cells they're just not just a conduit there, like to transport lymph. They are there because they also secrete a lot of factors. And when tumors are so very aggressive, they can also remodel the immune system. And this, I will talk about that later. So the immune system, of course, you know, is our defense, or the defense mechanism. It should be there to attack the malignant cells. And when the tumors are not so aggressive, this immune system can win. But in aggressive situations, so really what happens is a rewiring of the immune system. So instead of attacking the tumor, favors tumor progression. And I'm going to talk about this, how to do that. So what we wanted is to have, so we knew from the literature that early stages of melanoma development involve the expansion of the lymphatic vasculature. This was known. What we wanted to see is the process at the whole body level. And so what we did is to generate animal models for in vivo imaging. So, and because we wanted to pursue lymphangiogenesis, so first question was, what did you target in terms of imaging process? And so there are many receptors, many ligands, many receptors involved in the lymphangiogenesis process. Some of you will know, I mean, FGR, c-MET, IGFR. These have also other roles in other pathways. That's why it's not that simple to identify pathways that will be more selectively related to lymphangiogenesis. Then there are also multiple effectors downstream of these signaling cascades. So in the field, so there was a lot of emphasis put into a factor is called PROX1. That's a factor that is involved in the transactivation of many lymphangiogenic factors. So what we chose to do work is on this axis, so particularly this receptor VEGFR3, which is the receptor for two main ligands, VEGF-C and VEGF-D. I'm going to tell you now why and the two things. So PROX1 was and had been used in the field because it's a protein that is expressed very early on in embryogenesis and then stays in the adult. And then so this is used to now the promoter of this gene to drive GFP and other fluorescent factors. And this was very useful in the field, particularly to visualize this adult, the lymphangiogenesis in the adult. Now, VEGFR3 is a bit different, and it's different because in the adult, the expression is actually downregulated, except when you have pathogenic situations, such as cancer or even inflammation. And this induction is what allows for the window opportunity to really have a way to very more detailed way to quantify and to visualize the lymphangiogenesis. So Sagrario Ortega at the CNIO, she was already working and she was generating a series of knock-in animal models. And then when we joined the CNIO, as Santos said, in 2008, a postdoc in my group, David Olmeda, got into modifying these animals for melanoma. So what these animals are, so it's a system where we introduce by a knock-in technology, so GFP and luciferase cassette. under an internal ribosomic entry site. And this is inserted in the untranslated 3' untranslated region of VEGFR3. This means that VEGFR3 is going to be expressed normally, and then coupled to the expression transcription of VEGFR3 is going to be the expression of GFP and luciferase. Okay. So what we follow really is using a medium way to monitor changes in the VEGFR3 expression. And this allows by GFP, you can see here the vessels, and then more importantly, let me take this out of the way so you guys can see. So you can see, so I mentioned in adult mice, very minimum expression, and then when you have a tumor, you see now these animals light up. And so we could do many things now, yeah. So we can work, we have immune competent and immune deficient versions of these animals. The immune deficient we can use to implant cells, and we can implant now tumor cells, human mouse have different tumor types, and then we can ask the question, so we can express or inactivate genes. And for us, it was so interesting to see that so this is the site of implantation here. And when the melanoma cells were very aggressive, so we could implant, and already before even seeing lymphangiogenesis in the implantation site, you could see it systemically at the draining lymph node, the contralateral sites, and other organs. So a systemic activation of lymphangiogenesis. This is important. I told you that melanomas from very early on, thin lesions can metastasize, and then we could see in vivo that indeed these cells have very early on these systemic effects. Then we can also implant patient-derived biopsies, and then we can play also with different aggressive behaviors. And again, so when the metastases are aggressive, activation of this process. And we also have now genetically modified mice. I'm not going to get into all the details, but these animals develop melanomas driven by oncogenes in these tumors, the BRAF, and then in the presence of PTEN, so the inactivations of tumor suppressors. And also you see how when the tumors grow, and then you have this systemic activation of neolymphangiogenesis, VEGFR3. And this is now, you can imagine it, now this is a tool. This is a tool because we can collect and address at different times. The animals are alive, we can follow them, we can decide when and where to now go for what we think is a pre-metastatic niche. And we know there's a pre-metastatic niche because we can do things as what we do in the clinics. In the clinic, where these are melanomas here, you see, and how do they emit? The animals emit luciferase. And so the question for us was, now what happens if we remove the tumor at the implantation site? Are these signals going to be still there or going to disappear? And this was important because if they are there, means that you already have tumor cells for already probably mounting a response. But if they were driven by the tumor, I think I lost the. So then it's like to see to which extent the primary tumor is sending or actively required for sending these signals. So what happened was this. So we removed by surgery the melanomas, and I was so interested to see that most of the signals went away. So that means I said that how important the tumor is to favor this distal activation. And then what happened is like in the clinic, so if you wait when melanomas are aggressive, so now there is relapse. And we can see if we didn't have the animals, we had to wait a long time. So you see here metastasis is pigmented and non-pigmented. But very early on, so before we could detect it visually, so our animals were already lighting up. And they were lighting up on the sites that they will go and metastasize. So that's why we call these animals meta-alert, as a way to now follow the metastatic and gauge the metastatic potential of cells and tumors. So long story short, so using these animals, I mentioned that we can work with cells, we can work with patient-derived xenografts, and also with genetically modified mouse models. And then the question was, okay, so what are the signals that favor very early on, before we could detect tumor cells in these sites, what are the signals that get activated in these pre-metastatic niches? So we did a lot of studies involved proteomics, indeed. We started first with cell lines, and then we moved into tumors from patients. So one of the proteins that we found here is in red, so this is expression. One of the proteins that were most overexpressed with in these settings where we have this distal activation of metastasis was a protein called midkine. But midkine is a growth factor. And we picked it because it was found to be overexpressed in many different tumor types. And indeed, this protein is secreted and has been used to monitor, for example, breast cancer and metastatic load and so on. It was interesting because it's a ligand, it's a sticky ligand for many receptors. And integrins, ALK, NOTCH, LRP1, and these kinases. And it was known in the field that there were various roles in metastasis, in chemoresistance, and aspects of apoptosis, but was not known. Firstly, there had not been data in melanoma, and secondly, there were no data in the context of lymphangiogenesis, so we thought it was an opportunity for discovery. And so I'm going to just summarize a lot of data that we had, because, and so first thing what we did is the usual, so start with cell lines and deplete this control. We depleted midkine by shRNAs or by CRISPR strategies, so genetic depletions. And what happened is that the tumors you see here at the implantation site still grow, but you don't see animals lighting up that much. And indeed, what happened is that there was a reduction on metastasis here, a lymphatic lymph nodes, and also we have reduction of distal metastasis. We overexpress the protein, it's the other way around. Here we have control, this is a truncated, so the full length, sorry, and this is a truncated form. And it was interesting because you see, this is a cell line that you see there, that generally we have this tumor, it's actually a big tumor, but it doesn't have the potential to metastasize, and just overexpressing midkine, and we had lots of controls to know the physiological levels, so we could see now this activation of VEGFR3, lymphangiogenesis, metastasis. These were in mice, so of course we have multiple collaborators, clinical collaborators, and for us it's of course essential to demonstrate function in patients. So here's immunohistochemistry. In pink, we are staining for midkine, and you see in EVI very little staining. And then as the tumors progress, you have more and more staining. This is the quantification. And it was not only that there was more stained, is that the patients had different survival. This is a survival curve, and what you see here is how the patients die depending with time. And here, if they have high levels of midkine, they die a lot more than those patients that they have low levels of midkine. And so with this, we concluded that we had identified a protein that is secreted by, so here's the summary. It's a protein secreted by melanoma cells. I don't have time to show you, but we knew that this accumulates in the context of the vasculature, lymphatic vasculature, and through mechanisms involving mTOR, this transactivates VEGFR3, and this is sprouting more lymphatic vessels there. Of course, for metastasis, it's not sufficient that you have more vessels. The cells have to move, and they have to intravasate, and they have to get in. And we found also, and we described in the paper, a couple of years ago, some years ago, that midkine also favors the motility of melanoma cells and all of this intravasation. And this was kind of the first group of studies. And so when we started working with a protein, so when you work with a protein that is secreted, that has receptors that are expressed by various cell types. So the question is, are there other cells that could be influenced by this? Well, sorry, this is my Torre de Hercules. Now you understand why we call it a Torre. It's our imaging tool. So I was mentioning a role in the immune system. Not going to get into this for if those of you that are working in cancer, immunotherapy is now kind of the theme. It's been tried different aspects, but compound or antibodies that block checkpoint inhibitors. Then, so of course, oncolytic viruses, vaccines, many different strategies. And it's working to a point. And for example, in melanoma, we can get now from, this is here says 40, but nowadays we have patients from 50–60% of melanoma patients responding to immune therapy. Still, whether it's a long-term response is something that's been analyzed. Also, there are lymphomas, there's lung cancer, there are many different tumor types where immune therapy is being checked. Now, in the context of immune therapy, something to keep in mind, so how complex the immune system is. So usually the patients that respond, or the tumors that respond well, they do because they recruit immune cells. Here you have them in various immune cells, CD8 T cells, cytotoxic T cells, they recruit macrophages, and cells that they do, and dendritic cells that they present antigens, and then they kill and remove the tumor cells. The non-responders are a bit different, so you can have non-responder patients and tumors because either the immune cells are excluded, so they cannot get in, or what they call this like ignored. So really they are just, so the tumor is silent. And this is what they are called cold tumors. This is a hot tumor. This is a cold tumor. And then there are other groups of tumors that are much more complex. And these are tumors that they are inflamed in the context that they do have. So you look histologically, you would see a lot of immune cells there. And immune cells being a lot of macrophages. But then they have also suppressive cells, myeloid-derived suppressor cells, and other cells that are rewired. And indeed, what you have is a suppressive infiltrate, okay? And so there's a lot of interest in distinguishing the different kind of modes of activation or remodeling of the immune system. And I'm going to tell you that Midkind actually what does is works on this and getting an inflamed but immune suppressed tumors, which are even more aggressive because these tumors are using the secretome of the immune cells to actually favor the aggressiveness. So we got into the immune system, just addressing the downstream signaling cascades associated with Midkind. So here you have, this is a study, so heat map. So this is a study on the Cancer Genome Atlas data set. Is 450 melanomas, and we analyzed the RNA-seq. And then we had identified so two group of patients that, the pointer, okay, that recapitulate, so we call signature, they just have low Midkind associated signature, and then the patients that have high. And these patients have a lot of alterations in the immune system and other pathways that favor tumor progression. But in terms of the immune system, it was interesting because this Midkind high associated signature patients, they have alterations in innate immunity, adaptive immunity, cytokine signaling, and hematopoiesis. So for us, this was very interesting to see how we could just based on Midkind and the Midkind downstream effectors, we could separate patients with different immune profiling. Indeed, then we look into this in more detail. These patients, and here you have low, high. Low, high means this Midkind associated signature. So they express high levels of TGF-beta, which is characteristic of immune suppression. They have lots of macrophages, but macrophages in a pro-tumoral mode. We looked at, and these are the patients enriched for these macrophage signatures. We stained in tissues, and then you can see here in the bottom, so staining in pink for Midkind, and so staining for markers of macrophages, C163, and they do have more, okay? So more Midkind, more macrophages in an immune suppressive background. So then what we did is a lot of analysis to assess the function of, so mechanistically, so what's happening here. And so for those who didn't, so there are many ways you can identify macrophages by flow cytometry. There are different markers. Here you have some. One of these marker characteristic in mice is a protein called arginase 1. When they have arginase 1 positive, they are tumors associated. So they are so immune suppressive. and indeed protumoral. This is an example of how we can visualize this process in these macrophages. For example, here you have animals where we implanted melanoma cells that do not have and melanoma cells that do have MITK. These are isogenic cell lines. And then we stain for various things. So DAP is the nuclei, MITK is in green, you see here extracellularly. And then this marker arginase 1, you see the difference. Lots, it was a massive infiltration of cells that have this arginase 1 positivity. Then with all the various markers that you can do for flow cytometry, we defined that were myeloid-derived suppressor cells, and in particular, and more so, tumor-associated macrophages. And now I'm going to just summarize a lot of data, but maybe just for some of you that you might be interested in macrophage signaling. So how do we define this process? So first thing, what we had is that we looked at, so we took melanoma cells that express MITK, we looked at macrophages, and we demonstrated that indeed MITK so will increase transcriptionally the activation of this protein arginase 1. We look at the secretome to identify, so how was that? So what is so being also activated and in this process, and because it's very important to address the microenvironment of these macrophages. And then we identify here, this is like a lot of proteins. So here, oh, this is going automatically, I think. So TGF-beta and a lot of secreted factors, which together we demonstrated what they did is, so macrophages, part of their action is when they are in a protumoral mode, is to interfere with CD8 cytotoxic lymphocytes. And what happens here that we found that they are dysfunctional. They express a lot of markers that are associated with dysfunctional T cells. Dysfunctional CD8 T cell, what it means that instead these cells, their function should be to kill tumor cells, and what they do is they do not kill it because they have a lot of suppressive signals. And then what you have is this situation that you have tumor cells that do not die. And then, so what when you have T cell dysfunction, there are different signatures that different authors have related and addressed in the context of response to immune checkpoint blockade. And here you have in red, so patients that they have this dysfunctional, I don't know why this is going automatically, but sorry. So we found that patients with this MITK high signature in red, so they had features of non-responders to immune checkpoint blockade. So then we decided to address this functionally. So now what we did is to compare the effect of the response of tumors with or without MITK to characteristic immune checkpoint blockers, and this is particularly anti-PD-1. So what you have here, survival, these are now tumors in mice. So if they have MITK, they die very efficiently. If they don't have MITK, they survive more. But what was important is now when you treat with this immune checkpoint blockers, for example, this antibodies against PD-1. So now we have, it was very nice to see how there's an extension of survival of these patients, okay? So with now this, we offers now the possibility of using MITK and indeed MITK inhibition to interfere with the response to immune checkpoint blockade, which is very actively pursued in the clinic. We did more work to identify what was the signaling cascades, how was this MITK and immune checkpoint blockade helping each other. So these are heat maps, so basically differential gene expression. And what you see that if the tumors do not have MITK, and on top of that, they are treated with anti-PD-1, this is now in red. So we have a lot of signaling cascades involving interferon, anti-tumoral macrophages now, and T-cell infiltration. What is the conclusion of this? The conclusion of this is that when you have MITK, you have the tumors in a mode that are immune suppressed. If MITK is not there, so you have now a shift in the microenvironment. The microenvironment moves from TGF-beta to interferon, priming, and now the cytotoxic T cells can be activated properly and then can kill efficiently the tumor cells. And this is very important because this is actually being actively pursued in the clinic. And more that, so in terms of the clinic, what we did is now, because there are many clinical trials ongoing with immune checkpoint blockers. So what we wanted to see is that if the signals and pathways we had identified linked to MIDKIND were or not enriched in these patients that we were treating. Bottom line is that they did, these are complex figures, but the summary of this is that so the patients that, so basically they recapitulate the signatures that we have when we block MIDKIND and we treat, and it's like jumping on its own. So basically we have an enrichment of, we can predict, let's put it this way, response against anti-PD-1, gamma interferon, and then other combinations of anti-PD-1 and CTLA-4, which is other factors that people are pursuing in terms of the clinic. And here are different data sets that we used. And I told you about melanoma, so this data, then we looked at other tumor types. And then we have found that basically this activation of immune suppression, here I show you TGF-beta or macrophage signaling. We found it also associated with MIDKIND in lung squamous cell carcinomas, in glioblastomas, and also kidney renal clear cell carcinoma, among other tumor types. And this is now the summary. And now I'm going to stop a minute, so just to recap on what I have told you. So this is actually this is a drawing of a mother of the first author. She is an artist. And then when we told her about the immune system, this is how she envisioned the immune system. This is what we told Nature for the cover. So Nature Medicine. So this is like the picture of a macrophage, the way she put. And then when MIDKIND is present, so these macrophages are in a mode involving NF-kappa B, TGF-beta, and so that actually favor tumor progression and favor resistance to immune checkpoint blockade. And then when we block it, we have the other way around. Now we have these same cells that are there, and this is important, that they're in the tumor. Now they can be shifted into a killing mode, and you see cells here dying. This is the way she understood the dying. And then so that is offering the opportunity for a combination of MIDKIND inhibitors and immune checkpoint blockade. And then we have identified also markers to follow this, to stratify patients in the clinic. Okay, so I'm not sure how much time I have because we started a bit late. So I'm going to take 5 minutes or so. So because I just wanted to many maybe of you here and the audience might be interested in treatment and how can, so the first part was gene discovery, now is treatment. And of course, when, but one thing, a disclosure, so I'm going to talk about a compound that is was developed into a company, Bioncotech Therapeutics. Now having the animals that emit light, now we can use them to see compounds that they turned off this light, okay? And we tried various, so immune checkpoint inhibitors, BRAF inhibitors, so different small molecule inhibitors. And then we have our own compound, which I'm going to get into this, BO110, actually very efficient. With one dose, we can see how the signal of this lymphadenogenesis go away, and then you treat more, the tumors go away as well. And this is in mute mice and immune competent animals. For those of you interested, you can imagine your tumor type and you can imagine your favorite drug or antibody or compound and see if it works, if it doesn't work. In our case, it was very nice. This compound we had published some few years ago that it was an agent based on long double-stranded RNA packed in a way that got inside the cells, and this induced a form of death. So I have here, so this is forms nanoparticles, this is electron microscopy, you can see them. It's sensed by a series of double-stranded RNA sensors. forms these vesicles, activate, and the cell basically auto collapses by autophagy and apoptosis. And this is what we knew before we got started with this, the animal models, the meta-alert. And then with the meta-alert, now we had an added feature of, as I showed you, inhibition of lymphangiogenesis and metastasis. And this was the basis of this company, Bioncotech. Now, so having the animals, and I told you that we have effects on MIDCAN, we have effects on beta-FAR3. So now we have ways to pursue the effect of this compound BO110. And what we found is indeed that it shuts down both this protein and then the vasculature involving the lymphangiogenesis. And so there are so many assays that we do to define this repression at the transcriptional level, etc. And mechanistically involves activation again now of signaling cascades involving the immune system that, so in this case, are mediated by interferon in this type 1, and interferon alpha and beta. What is important? It's important because the interferon is the main modulator of the immune system in a killing mode. So for CD8, CD4 T cells, monocytes, macrophages, and dendritic cells. And indeed, we found that what happens now is that we can have the tumors I told you that they were cold or deregulated into hot. So basically, we have a compound that by chance, by looking at this mouse, by finding that had effects on the tumors, on the vasculature, and then on the interferon, we found that actually promotes this levels of all these immune cells in the tumors that are recruited. And to our satisfaction, so now Bioncotech, this is merit of the company. Now the company is highlight, so they changed the board and what's got merged into, so what Highlight Therapeutics, and Highlight Therapeutics has now, this is a patients, here they have a tumor when they are treated, and these are very aggressive melanoma, patients with aggressive melanomas that have not responded to other treatments, and they are used in combination with immune checkpoint blockade. This was already like a few years ago. Now they are treating patients of different tumor types, and it's like the phase 2 clinical trials. And to me, when I saw this, this was presented in ASCO, was one of the pictures that, you know, that it was not my data, but it was our data, but this is just in the patients. And it's so happy when you see something like this. Of course, there's still many studies to be done, but so far it looks so good. And that's it. It's just what I wanted to tell you. So how we have our systems to visualize melanoma, metastasis, how using these animals and patient material we can find new genes, and how we rewire the immune system, and then how can we use this for treatment. And with that, so this is a lot of people in my lab involved in these studies, the funding agents, so was, well, David Olmeda and Daniela I have to mention, but a lot of we and collaborators. And because it's late, I just so wanted to finish with this. And I told you that Margarita, for me, was important on many different levels, but also one that I appreciate a lot and appreciate more and more right now, indeed, was this concept that for her, all men and women in her lab, students and so on, postdocs and seniors, were treated the same way with the same, so basically all of us, what we have to do is to work and get our data. And for me, this concept of normality, of being just a scientist, she even never, when it was there, so it was not an issue of you can or you cannot. For her, it's just you can. And that's it. And it's something that I know that I coordinate, and I have this group in ASECA Mujer, the Asociación Española de Investigación sobre el Cáncer. I want to finish with this because I think to me was also the point that, yes, we can make it, and the women as well, and some women here, and I know that some of you are out there, so keep going. And so that's it. Now I can take any question, and I'm glad to answer. Okay, thank you so much for the invitation. maybe I should stop sharing.
Thank you, Marisol, for this amazing lecture. Something about the questions, or if anyone in the room wanted to, to ask questions, feel free. And also for the people, following the session by Zoom or by the YouTube, just write the questions in the chat, and I'll try to take care of that.
Okay, yes.
Well, thank you, Marisol, for this wonderful talk. Impressive results, very clearly explained. I just wondered, is there anything known about what regulates MYC expression in vivo or its secretion?
Yes, the question is, so in terms of MYC, so what is upstream? So how is it regulated? This is a very important question. I get this every time. And, we're working very hard on this. So we know, so this is a growth factor and has been described, regulated by hypoxia, by estrogen, so different kind of stress signals. In our case, we know that, so there is part of PI3 kinase, so a series of feedback loops that are important for the regulation, and we're working very actively on this. So there's not one single driver, let's put it this way. So we know that, and also different cell types, we're working with melanoma cells, and I told you that the other cell types that they have also MYC associated signal in there. So there's, so we think upstream, there's definitely so oncogene-driven effects by the PI3 kinase particularly. And then we have ALK-related, not related feedback loops. But we still have to define the receptor. We still have to define other things.
Question? Actually, I have one.
Yes.
You have mentioned that the presence of MYC in several tumor cell types. One more question is whether it is also present in normal proliferative cells, and to what extent is the regulation, and also related to the control of expression. What do you know about the transcription factors, so even the chromatin landscape in the tumor, so for that to influence the expression?
Yeah, this I didn't get into this. And so MYC, the name of MYC comes from mid gestation. So it's a gene that is essential for embryonic development. And so the reason why we got into this, and maybe I should have mentioned, so is that the expression in the adult is actually very much downregulated. With some exceptions still in the ovaries, but this is more, it's kind of shut down. And indeed, we have now generated another reporter animal. So so I told you about VEGF3 reporter. Now we have a MYC reporter, and we can see beautifully how during embryogenesis, so there is activation in many different areas related to the bone marrow, related to the reproductive system in the brain, very much expressed in the brain. This is known, and then it's shut down. So why and how is it that inhibited and is inhibited during development? This is not known, it's not clear. So for us, so the question was what is activating? For us, it's actually more interesting what is suppressing the activation in normal settings. And then the other issue is then how the tumor cells. The tumors is because, and that's why we always look a lot at the secretome. Is a secretome of, so the context of the tumor is actually what drives the activation of this protein. But it has a function in development, very important, brain development and cardiac development as well.
Any further questions from here? So I don't see any questions coming out from the chat, so if there are no, how about it?
Understood. So in this system, you are labeling, so you are labeling this as pre-metastatic disease. So something where this lymphadenogenesis is being boosted, but there is no metastasis there
yet. No. Yes.
So it's not like it's not MYC reaching there or anything like that. It's somehow the effect.
No, MYC is reaching there. So maybe this I didn't explain the whole thing. So no, MYC is secreted, and then we see this protein. The reason why we also pursued is that when we look histologically at these sites, when you have neolymphangiogenesis, we see MIDKINE in there. So the protein gets there. And this is the active being there is when then drives the activation and secretion. Not only MIDKINE and factors associated with the secretion of MIDKINE.
It's not the whole body.
No, that's very interesting. There are many sites that are prepared, but there are some that get colonized. And it's like, I always tell the people, it's like when you have a party and you invite different peoples to different rooms, and then the rooms are prepared, but then the cells have to reach there. And so the cells that reach to the different sites, that's a different situation. So we always see neolymphangiogenesis preceding metastasis, but there are some sites that do not get colonized, and they may not get colonized because we don't follow them time enough, or because the cells did not reach yet there. But we know that they reach, so there is activation before the cells get there, because we did a lot of analysis to prove that they're not cells by PCR and histology, that they're not there yet. Yes.
So MIDKINE modifies the responses of macrophages to a tumor. Mm-hmm. Is it working as a macrophage attractor, as a chemokine?
Yes, the question is now, so I told you in terms of, so the effect of MIDKINE on macrophages. Yes, it does. And so it has several levels of action. So one is definitely a lot of chemokines being produced, so macrophages are there, but it's not just that they are there, then they shift. And then is when they acquire, I told you this arginase 1 in mice, but then in human cells is the shift in the transcriptional program. So the macrophages, instead of acting as antigen-presenting cells, that's where the function should be, and also killing and digesting the tumor cells, what they do is they support the growth. They switch, the transcriptional switch.
Okay. So if there are no more questions, I think that we can finish the session. Thanking you again for this.
Thank you to you.
In the memory of Margarita Salas. So thank you, Margarita.
Okay.
Fantástico.